This conference will now be recorded.
PPTs. So some few PPTs are there in a very simple manner, and I have asked them to be uploaded onto the website, and you can go through those PPTs in very simple points. The things have been given. Please go through them. In the meantime, today's class is about virus-host interactions, and we limit ourselves to that. But we'll first see a short video on how actually viruses replicate or try to enter the cell. So as you might be knowing how COVID is entering the cell, all the details you might have read. So we'll be discussing from time to time. We'll be discussing about some examples of viruses also. So in the meantime, let us see the video. So I hope you understand the very basics of virology. The main thing is viruses can either be DNA viruses or RNA viruses. That is how they are classified, because viruses do not contain both DNA as well as RNA. They are either DNA or RNA viruses. That is one very basic fundamental. Another fundamental thing is they are intracellular organisms. They have to stay inside some cell because they do not have the essential requirements for their own reproduction, for their own replication. So that is why they instruct, as you saw in the video, they instruct, or in fact they hijack the host cell's machinery to make their own virus particles. Okay. So those are the very basic things. So please go through the first two chapters. Virus host interactions is given in the second chapter. The first chapter is in fact very, very important. The first two chapters, once you understand, you can understand the rest of the virology chapters. If you do not understand the first two chapters, it will be a little bit difficult for you to understand the other things related to the particular viruses. So that is why the first two chapters are very, very, very important in virology. Whether you're reading Anantanaran, whether you're reading Baveja, doesn't matter. But you have to read, you have to go through the first two chapters thoroughly. Okay. So because we are concentrating on virus host interactions today, so we'll discuss basically what happens when a virus enters inside your body. Okay. So what are the possible outcomes of viral infections? Either it can be an acute infection or it can be a persistent infection. So that we already know. It is not only true for sorry. So it is not only true for viral infections, it is true for any other infection also. So either it can be an acute infection or it can be a persistent infection. So persistent infection is also of many types. Either it can be chronic or latent, or it can transform the cell. I hope you understand what is meant by transformation. So transformation means, like, suppose a virus has entered and it is an oncogenic virus. It will transform your cells in such a way that they may become cancer cells. Okay. So that is transformation. So that is one word you have understood today. So transformation is changing the cell in such a way that it can be a cancer cell. At the same time, some sort of persistent infection can be a latent infection that we see in the case of herpes. I think you might have seen some people, they'll have small vesicles whenever they have fever. They are also known as fever blisters on the side of the mouth, etc. So those are, herpes is a latent infection. It will remain latent inside your nerve ganglia, and from time to time, suppose there is fever, suppose there is some stress, suppose you're exposed to sunlight, they will be somehow, they'll be triggered in such a way that it will again cause the disease. The virus does not leave the body, but it is latent or lying just sleeping for the time being, and again it will again activate and it will cause the disease. Okay, so that is known as a latent infection. Then there is a chronic infection. So chronic infection also, you might have seen some infections for a very long time, they will persist inside the body. They may or may not cause symptoms, and they, from time to time also, they will cause symptoms. So that is acute infection and persistent infection, and the different types of persistent infection which happens in viral infection. So chronic, latent, and transformation. Then there are symptomatic infections or inapparent infections. Symptomatic means you will actually find either there may be fever, there may be some sort of flu sort of symptoms. So there may be symptomatic infections or there can be inapparent infection. In the parlance of COVID, you might have known some people, they might be infected, but they are asymptomatic. So you might have come across the term asymptomatic a lot in these past few months. So that is actually an inapparent or an asymptomatic infection. So even though the person is infected, we may not know it because the infection is not apparent to the outside. He will not have any fever, will not have any cough, will not have anything. So you will not know that actually they are symptomatic or they are having some sort of infection inside. So the outcome of a viral infection depends from the characteristics of the virus-host interactions and from the host defense response. So not only does the virus become important here, but also the host defense response. So you might have come across the term herd immunity. Yes, so many people are talking of herd immunity. Why herd immunity is so important in COVID? Why our own immunity is so important in COVID? Because the way the virus is, it will interact in some way with the host, that is the human being, and the way our own immune system or our own defense response reacts. So the ultimate thing which is visible outside, it will be a mix of both the virus as well as the host defense. So determinants, there are different determinants of viral pathogenesis. There are different determinants of viral disease. So viral pathogenesis, so there are different viral factors, so virulence factors which will be there, so which will cause the pathogenesis, and then ultimately causing the disease. There will be different determinants. So ultimately it will depend on the virus host interactions. So how does the host interact, how does the host's defense response react, and then how does the ultimate response determine what actually happens. So you can see that there are actually many determinants. So we do not want to go into that much detail, but we'll just know what are the points. So first thing is, what is the interaction with the target tissue? For example, there are some hepatotropic viruses. Like for example, the you might have seen some hepatitis viruses like hepatitis A, hepatitis B. You might have come across that. In spite of whatever route they may enter through, so they may enter through the mouth, they may enter through any sort of parenteral route. So, but they will ultimately target the liver. So ultimately, what is the interaction with the target tissue? What is the access with what they get with the target tissue? What are the presence of receptors? You might have come across in COVID. So there are certain receptors in our respiratory epithelium, which the spike protein or the receptor of the virus which it interacts with. So that is the interaction with the target tissue. Then there is the ability to kill the cells, that is called cytopathology. So there will be some sort of viral replication within the cell, and in some cases after viral replication there will some cells may be killed. So there will be cytopathology. So there will be cytotoxic viral proteins, there will be inhibition of macromolecular synthesis. So because hijacking is there, so that is one factor. So the ability of the virus to kill the cells. Then there is host response to infection, that is how we react to what the infection is. So our intrinsic cell response, our innate immune response, our acquired immune response. I hope you remember what the innate and acquired immune response was. So innate is what we are born with. Yes, what I have got as being a child to my parents, what I have got as by virtue of being a human being. So that is my innate immune response. Acquired immune response is what I acquire. It may be through infection, it may be through vaccines. So whatever I have acquired through my life throughout my lifetime. So that is acquired immune response. So all these determine the host's response to the infection. So how the infection will come and then how we will react. So both sides are important. Then there is immunopathology, that is our body will produce interferons, there will be T cell responses. So all these factors, our antibodies which are produced, the complement which is there, the immune complexes which are formed, so all those are also included. Then there is ultimately the nature of the disease, that is the target tissue, the site of entry, from what site it has entered. For example, rabies, it has entered through some dog bite, but how it has gained access to the target tissue, that is slowly somehow it has gained access to the nervous system, and then slowly it has ascended up the nervous system, and then it has ascended up till the brain, and then it has caused some sort of pathology. So that is the target tissue, the nature of the disease. Ultimately, it will depend on the site of entry, the ability of the virus to gain access to the target tissue. And in the case of rabies, when you come to the chapter, you will see that suppose the bite or some sort of wound which is there and the virus has entered near about the place where the brain is. There's for example, it is on the face or somewhere. So naturally, the pathogenesis will be earlier. Okay. Whereas, suppose it is on the leg, then there is naturally it will take some time to gain access to the tissue. Then there is the severity to disease, that is the ability to kill cells. Then the immunity to the virus, the intact immune response, immunopathology. So all these play a role in how the virus will interact with our body, and then how our system tries to control that viral infection. So what are the hallmarks of virus-host interactions? So we will go through in a little bit of detail. What are the determinants of viral disease and nature of the disease? The type of target tissue, that is the replication sites. The pathways of viral entry, viral spread to the replication sites, viral tropism. You might have come across this term tropism. Tropism means it is sort of an affinity for particular tissue. For example, we were talking about the hepatitis viruses. So, irrespective of whatever route the hepatitis virus has entered through, it might be a fecal-oral route, it might be a parenteral route, but ultimately it will go to the liver. So, that is a hepatotropic virus. So, similarly, the rabies virus, irrespective of whatever route it has entered through, it will ultimately gain access to the CNS. So, that is known as viral tropism. So viral tropism is where it will ultimately launch. And then the cell permissivity to the virus replication and the virulence of the viral strain. You might have come across in COVID, especially. Why I'm giving examples, because you might have read a lot of things about COVID, and you might have understood a lot of things about COVID, and many people, your own relatives, your own friends might be asking you about COVID. So the virulence of the viral strain, for example, so many different strains, you might have heard that at least few strains of COVID are there. So it depends on what sort of viral strain is affecting the person. Okay. So the viral strain is also important. So each and every strain is different, and then it depends on the virulence of the viral strain. Then what are the other determinants of viral disease which causes the severity of the disease? One is the cytopathogenic attitude of the virus. That is how much amount of damage it can cause. Just now we discussed that how much amount of cell killing or cytolysis it can cause. What is the immunopathology? What is the initial inoculum of the virus? So suppose initially a very huge load of the virus. You might have heard about viral load in the parlance of COVID. So suppose the viral load in a patient is very high and you have come in contact with them. So naturally the chances of your contracting the disease is very high. So naturally, so the inoculum of the virus, so initially how much amount of virus is entering into your body, so that is also important. Then it depends on the compromised host. So suppose the host is on steroids, suppose the host is already having HIV, suppose the host is having some sort of transplantation and he is on immunosuppressive drugs, so naturally he will have more severe disease. Then there is the host genetic background. So suppose some people genetically they may be immunocompromised, and the age. So suppose the age is either in the extremes. So suppose either it's a baby or it's in the older age group. So naturally the chances of contracting the disease or the severity of the disease, it will be. Then there are other determinants like the host immune conditions, like the competence of the immune system. So how much amount of competence our own immune system is having, and how our own immune system is trying to control the disease, and how much amount of previous exposure to the virus has occurred. So suppose previously I have been exposed to measles, I have had measles. So naturally, even though I am exposed to the virus, if I have immunity, naturally, excuse me, so naturally I will not get the disease. So previous exposure to the virus and what sort of immunity, because you might be remembering about memory cells. Yes, so memory cells are there, T memory cells, B memory cells. So those memory cells will be there, which will remember that this particular disease had come in earlier. Okay, so that is a part of our immunity. Then what is the viral pathogenesis, or the process by which the viruses cause disease? Okay, so first thing is the viral entry. Okay, as we saw in the video, you will see the first thing is the viral entry, and then there will be viral spread. So from the initial entry point, it will spread to other places, and then there will be tissue invasion. Some sort of tissues will be invaded, and then there will be tropism. Just now we were discussing about tropism. Tropism is typical affinity for a particular, if it is having affinity for the CNS, if it is having affinity for the liver. So it will ultimately, whatever route it has entered through, doesn't matter. It will ultimately, it will have affinity for that particular tissue. So that is known as tropism. Then there is virus shedding and transmission. So ultimately, after the virus has replicated, then there will be virus shedding, and then it will be transmitted to other people. And there is the host defense, which is ultimately a important point in all this. So initial infection will lead to an incubation. So there will be an incubation period. So for some time the virus will try to replicate, it will try to spread all over, and then the virus will spread in the host, it will go to the target organs, it will cause symptoms, and then there will be the host response. The local responses can be there like the interferons, and the systemic immunity also can be. So all the things are important. So what are the sites of virus entry into the host? So there will be, you will come across so many viruses in the subsequent chapters. You will come across so many different viruses. There will be different routes of entry for each virus. So those things you have to remember. So it might enter through the conjunctiva, through the respiratory tract, the alimentary tract, the urogenital tract, the any arthropod vector. So any arthropod vector can be there, which can transmit the disease. For example, dengue, chikungunya. So these viruses, they will come through some sort of mosquitoes. Okay, so Aedes mosquito, it will come and it will spread the disease. So that is an arthropod vector can spread the disease, or it can spread through a scratch or an injury or through the skin. So there will be different routes of entry for each virus. So you can see that different routes of entry are there, like for example, the location, the respiratory tract, the localized upper tract or the localized lower tract. You can see so many different viruses are there. The rhinovirus, Coxsackie virus, respiratory syncytial virus. So some of them may enter through the respiratory tract, but they will follow the systemic spread. That is, it will go to different organs. For example, rubella virus, hanta virus, mumps virus, measles virus. They will initially enter through the respiratory tract, but later on it will go to other systems also. Then there are some viruses like the enteroviruses, which will enter through the alimentary tract. What are enterovirus examples? Like poliovirus. Okay, so poliovirus, fecal route it will enter, but ultimately it will cause disease somewhere else. So it is ultimately it will become systemic. Then some of the viruses will enter through the eyes. For example, enterovirus 70, these may cause conjunctivitis. So localized, like adenoviruses, they will ultimately stay in the conjunctiva. It will not go anywhere else. Okay, so then it can enter through the skin. For example, through arthropod bite. Just now we discussed about dengue and chikungunya, the Aedes mosquitoes. So through the mosquito bite, it will enter. So the flaviviruses, the bunya viruses, the pox virus, so they will enter through the arthropod bite. Or it may enter through a needle puncture, sexual contact, or animal bite. So animal bite like rhabdovirus, that is the rabies virus. So there are different routes of entry of different viruses. So then how do they spread? So finally, what will happen is it will penetrate, it will ultimately penetrate into the body and it will cause primary replication. So a very good diagram is given in your textbooks. Please go through your textbooks once you finish this class. So you can see that once the virus will penetrate into the body, it will undergo primary replication. This primary replication might be at the initial point of it might be at the initial point of entry, and in some cases it will be localized there itself. It will be localized there itself. Some viruses like the influenza virus and the enteric viruses, some of them viruses, they will be localized. Suppose they enter through the gastrointestinal tract or through the respiratory tract, they will be localized there itself. They will not go anywhere else. But in some cases, they will go into the blood and they will cause primary viremia. Emia is in the blood. So viruses will go and they will circulate in the blood. So that is known as primary viremia. Then they will go to the target tissues. Naturally, once it is going to the blood, it will go to the target tissues. So that it depends on the sensitivity and the permissivity of the target tissues as to how much amount of replication will occur in the target tissues. So it will go to the target tissues, and after it goes to the target tissues, it will undergo secondary replication in the target tissues. So it depends on how much amount of permissivity the target tissues are having. So this is known as secondary replication. And after secondary replication, there will be secondary viremia. There is again, it will spill over into the blood again from the target tissues. It will again spread over into the blood, and again it will circulate in the blood. That is known as secondary viremia. And then it can be disseminated. So, for example, exanthema viruses, polioviruses, it will get disseminated everywhere. Okay, so it will get disseminated in different organs. So, exanthema viruses, for example, you might have read about chickenpox. You might have read about the smallpox viruses. So, those viruses they will disseminate. So ultimately they will enter. So you can see this picture, it seems complicated, but actually it is the same thing which we saw in the flowchart. So ultimately it will enter the infection, it will enter through some sort of route. So it will enter through some sort of route. So it may be shed through that particular route. For example, herpes simplex virus or papilloma viruses, it will replicate at the site of entry. Okay, so whatever viruses they are entering or penetrating into the body, so at that particular place, wherever it is entering, it might be the skin, it might be the mucous membrane, it might be the respiratory tract, it might be the GI tract. So it will first enter into that, and then once it enters, it will go to the lymph nodes, local lymph nodes. Naturally, in the local lymph nodes also, it will try to contain, but at the same time, the virus will try to replicate, and then it will spill over into the blood. So this is primary. It will spill over into the blood. This is for the first time, so that is why it is known as primary viremia. So after primary viremia, but because it circulates inside the blood, it will go to different target organs. So it can be different organs. So in these different organs like the muscle, the liver, the spleen or the blood vessels, there also it will replicate. And again after replicating, again it will spill over into the blood. This is known as secondary. For example, hepatitis B virus, the polio virus, the immune immunodeficiency virus. So this will be the secondary viremia. For the second time, it is going into the blood. And then again, it will go to different organs. It will be disseminated to different organs. So with many organs involved, it will become systemic. So, for example, the skin, the mucous membranes, the lungs, the kidney, the gastrointestinal tract, the brain. So, different viruses will go to different organs depending on their tropism. So, for example, varicella zoster virus, that is the chickenpox virus. So, it will go to the skin. So, rhinovirus, it will go to the enteroviruses. It will go to different mucous membranes. Arena virus, hantavirus, these will go to the lungs. The influenza virus, measles virus, they will go to the kidneys. Then gastrointestinal tract, the rotaviruses, the enteroviruses, the reoviruses, they will go to the GI tract. And polioviruses, togaviruses, they will go to the. So depending on their tropism, they will lodge in different types of organs, and there they will cause their disease or pathogenesis. So these are the different sites of entry, viral entry into the human body. So here you can see this is through the respiratory tract, and once it is entering into the respiratory tract also, it is replicating. It is replicating locally inside the respiratory tract. So here you can see that the clinical manifestation might be there itself. For example, rhinitis or common cold. So the rhinoviruses, coronaviruses, the normal. I'm not talking about the COVID, but the other coronaviruses. So many coronaviruses are there, which are actually there, and we have at least once or twice in our lifetimes we might have suffered with coronavirus, common cold. So parainfluenza viruses, respiratory syncytial viruses, these will all cause rhinitis, pharyngitis, laryngitis. So all these things can be put. At the same time, suppose it goes further on, it can cause tracheitis, bronchitis, bronchiolitis. So all these viruses, other viruses like influenza virus, adenovirus, respiratory syncytial virus or parainfluenza virus, it will further go into different parts of the respiratory system. So then you can see that enterovirus pathogenesis. Okay. So enterovirus pathogenesis, you can see that here enterovirus, basically we are talking about, suppose some of the enteroviruses like polioviruses or viruses. So here you can see they will enter through ingestion or the gastrointestinal tract. They will come and they will replicate in the intestines, in the case patches. So there after that they will go into the blood and they will cause primary viremia. So after primary viremia, again they will go to the target tissues after secondary viremia. So here you can see that the target tissues might be different in different viruses. For example, polioviruses or viruses, they will go to the brain, they will cause encephalitis or paralysis. And then echoviruses or polio or viruses, they can go to the meninges, causing meningitis. Hepatitis viruses, hepatitis A virus, it will go to the liver. And the echovirus or A virus, it will go to the skin. It can cause hand, foot, mouth disease. You might have heard in children, some small babies, they will have hand, foot, and mouth disease. And then some echoviruses, they will go to the muscles and they will cause myocarditis, pericarditis, pneumonia, etc. So here you can see rabies pathogenesis. So a virus first, it will enter through the animal bite. So once the dog will bite man, it may be on the leg, it may be somewhere. So virus will first replicate in the muscle at the site of bite. So at the site of bite, it will first replicate. Then that is why it is so important that suppose you have got bitten by a dog, it is first important that because you have to wash that particular place with a soap and water. So because rabies virus, it is having an envelope. It is having a lipid envelope. So once that lipid envelope, you wash it with soap and water, that lipid envelope will dissolve. So that is why, like you might have heard about COVID also, that because it is having an envelope which is made up of lipids, that is why if you wash it with soap and water or if it's sanitized with alcohol, that is why it will help. So similarly, first aid if you are giving to some person who has been bitten by a dog is because it replicates at the in the muscle at the sites of bite. So that is why it is important that you should wash it with soap and or detergents. Okay. So then the virus will go to infect the nerve in the peripheral nervous system, and it will move by retrograde transport. Retrograde is it will move from downwards up. So from downwards up, it will go again. It will replicate in the dorsal root ganglion and travel up the spinal cord to the brain, and then the brain will be infected. And once the brain is infected, it will travel from the brain via the nerves to different tissues such as the eyes, the kidneys, the salivary glands, and it will lodge in the salivary glands. So another example is varicella zoster virus or the chickenpox virus. So first it will enter through the conjunctiva or the upper respiratory tract, and then it will replicate in the regional lymph nodes, and then it will have primary viremia in the infected T cells, and then it will replicate in the visceral organs, and then it will cause the chickenpox rash after subsequent secondary viremia and acute infection of the skin. Then latency, it will remain latent in the sensory ganglia of the peripheral nervous system, and from time to time, suppose the person is having less immunity or he's immunocompromised or he's having HIV, so from time to time it will come as in the wherever the particular nervous supply is there. So they, at that particular place it will come as in the form of rashes known as shingles, and that is known as herpes zoster. So that is known as herpes zoster. So all these you will understand once you go through each and every chapter. Just I'm giving you a few examples because we are discussing about different sorts of virus-host interactions in the body. So the viral pathogenesis is the sum of the effects on the host due to virus replication and the immune response of the host. So there are direct effects of the primary infection by cytolytic viruses. So ultimately the cytolytic viruses which will be there, they will kill the cells. So that is why there will be direct effects. For example, virus-induced lysis of the neurons in the CNS can be there by the poliovirus. And then there can be indirect effects by non-cytolytic viruses. Those viruses which do not kill the cells, there will be indirect effects. So, indirect effects like, for example, consequence of the immune response. For example, CD8 T cell mediated or CD4 T cell mediated. So, different viruses will have different effects. Even though they are non-cytolytic, they will cause indirect. So here you can see the measles virus. So it is first infecting, then it is causing primary viremia in the draining lymph nodes. Again, it is spreading to the entire reticular endothelial system. Then it is shedding, the virus is shedding everywhere. And then after secondary viremia, it is causing epithelial necrosis. It is spreading through all over the body surface. It can cause disease. It can cause Koplik spots in the gums. So all those things can happen. So there are different types of kinetics of virus replication. One is slow localized spread. That is very slowly it will spread locally. It can be intermediate or it can be fast and disseminate very easily, very fast. So acute infection, you can see general patterns of infection. For example, acute infection, it will come and again it will subside in a very short time. So that is an acute infection. Then there are persistent infections. So acute infection example is rhinovirus. You will get a common cold, you suffer for 2 to 3 days, maximum 5 days, then you will become ultimately all right. So then there is persistent infection. For example, lymphocytic choriomeningitis virus. So here it will persist for a very long time. So that is a persistent infection. Then there is a latent reactivating infection. For example, herpes simplex virus, which we were talking about, it will cause fever blisters. Whenever you are exposed to sun, you are having stress, you are having fever from time to time because it will be there in the sensory ganglia. From time to time, it will be reactivated. From time to time, it will show. Okay. So that is a latent reactivating infection. Then there are slow virus infections, for example, human immunodeficiency virus. So in the beginning, you will see a flu-like syndrome. Then for so many years, it might be there inside the body, but you may not know. And ultimately, it will again reactivate after so many years and it may cause death. So these are different types of infection: acute, persistent, latent, reactivating, and slow virus infection. So here you can see example is rhinovirus. You can see the acute infection. So it is a productive infection, it causes viremia, and then the clearance by the immune system is there. And common cold, we might have all suffered with common cold at some point of time. So that is an acute infection. So this is the course of a typical acute infection. So there is an establishment of infection, but because of innate defenses and adaptive immune response, what will happen is ultimately it will come. So then there is an acute infection followed by latent infection and periodic reactivation, for example, herpes simplex virus. So in the initial stages, there will be an initial productive infection with viremia. Here you can see there is an initial infection, and then after some time it will remain latent because it will be persistent in a non-infectious form, but it will be intermittently reactivated. From time to time, it will be reactivated whenever there is some sort of fever, there is some sort of stress to the body. So there will be reactivation, and then you can see that they will cause the fever distress, and then after some time again it will subside. So this is HIV-1 latency and reactivation because it persists inside the in a latent state inside the sensory ganglia. From time to time there will be infection by retrograde transfer. So again reactivation by anterograde transfer. So these are the productive infections of the epithelial cells. They will infect the sensory neurons, and then again from the sensory neuron again it will transfer, and again it will cause the disease. So then there is acute infection followed by chronic infection, for example HIV. So initially there is a productive infection with viremia, but then because the virus is not completely cleared by the immune system, there will be a continuous low level productive infection, and it will be a smoldering infection inside the body. So there will be initial infection where you will see that some sort of flu-like symptoms are seen, but it will persist for a very long time. It will persist till the patient dies due to some other disease. Okay. So these are the different types of infection. So initial productive infection of the permissive cells, then there is viremia, and then there is apparent latent infection, and then there is smoldering infection in the population of permissive cells. So a very slow chronic infection, this is very typical, which is seen in unconventional agents or prions. You might have heard about prions. So prions are infectious proteinaceous particles. So these are like scrapie or mad cow disease. You might have heard about mad cow disease. So these are continuous and slowly increasing production. So it may persist for 30 years, 40 years. So ultimately the patient will die. That is for certain. But it will persist for a very long time. It will persist for a very long time, for 30 years, 40 years it will persist, and ultimately it will be till the patient's death that the infection will go on persist. So these are the different mechanisms of viral pathogenesis. One is the cellular pathogenesis, one is the host immune response, and then there is the epidemiology, prevention. So first we'll discuss a little bit about the cellular pathogenesis. So here you can see there are different sorts of cellular pathogenesis. For example, abortive infection. Nothing happens even though there is initial infection with the virus is entered into the body. There is an abortive infection. So ultimately nothing happens. And then there is cytolytic infection. Cytolytic is there will be cell death. Okay? So the cell will die. So there will be viral production and cell death. And then there is persistent infection. For example, there is a chronic or a latent infection. So in those cases, you can see that also in transforming DNA viruses or RNA viruses, there may be immortality, cell immortality, or transformation of the cell. That is, the cell will change into a cancer cell. Okay. So these are the direct effects of cytolytic viruses. That is cytopathic effect, the production of inclusion bodies, apoptosis, and dysregulation of cell pathology, the cell physiology, and then host immune responses involved. So cytopathic effects, like for example, there may be different cytopathic. Cyto is cell, pathic is what sort of pathology is produced in the cell. So there may be inhibition of cell protein, host protein synthesis, host chromosome margination and DNA degradation. So nucleus shrinking. So all these things can happen. Okay. So the proliferation of the nuclear membrane. So these are the different types of cytopathic. So there may be production of inclusion bodies. So inclusion bodies is a short question for you. So there are intranuclear inclusion bodies or cytoplasmic inclusion bodies. Here you can see these are the cytopathic effects. So different types of cytopathologies you can see. This is for herpes simplex virus. So here you can see the normal cells are like this, and the cell which has the changed, that is cytopathogenesis has occurred. So here you can see that there is a change in the cell. So here you can see the cytopathic effects of Enterovirus 71. So in rhesus monkey kidney cells, so the cell has changed. So this is cytopathic effect. So here some cells there may be syncytium formation, that is a respiratory syncytial virus. So syncytium formation can be there. So many cells will fuse, so many different nuclei you can see. So that is a syncytium formation. So measles virus, respiratory syncytial virus, they will form syncytium. And then there may be inclusion bodies formation. So inclusion bodies, for example, reoviruses, they can form inclusion bodies, that is so many viruses, they will replicate together, and these inclusion bodies you can see through the light microscope. Normally the viruses we cannot see through the light microscope, we can see through the electron microscope. But these inclusion bodies, they will be so large because there will be a clump of so many viruses together, and there will be some sort of cellular pathology. So these inclusion bodies we can see through the light microscope. So this is Negri bodies. So Negri bodies also you will come across in rabies. So here you can see in the brain, you can see Negri bodies after the person has died due to rabies. You can see Negri bodies in the earlier days when there was no method in which they could diagnose properly. So Negri bodies are round to oval inclusion bodies which are seen in the cytoplasm, sometimes in the processes of the neurons of the rabid animals after death. So in humans also you can see the Negri bodies. So these are eosinophilic, sharply outlined, and pathognomonic inclusion bodies which are found in the cytoplasm of certain nerve cells, so in the brain tissue. So then we come a little bit about the post-immune response. Like for example, you know about all this, so I'll be very short in this. So physical barriers, chemical barriers, cellular defenses, innate immune response like fever, complement system, cytokines, inflammation. So these are all a part of our innate immune system. Then there is the dendritic cells, the macrophages, the adaptive immune responses like antibodies, the natural killer cells, the cytolytic cells. So these are the different types of defenses. So first line defenses, second line defenses, and the third line defenses. So there are different sort of defenses. Here you can see the intrinsic defenses, the innate immunity, and the acquired immunity. So ultimately the viral load will become lower and lower as they go through the defenses. So interferons and lymphokines, these are all questions for you. Okay, so please go through. These are short notes. So please go through these points. So interferons and lymphokines, they will cause fever, headache, and malaise, but ultimately they are supposed to control the viral infection. So delay type hypersensitivity, inflammation due to cell-mediated response. So all these will try to control the disease. So what is the epidemiology of viral diseases? Naturally, if the viral transmission is important, so it may be entering through aerosols. So right now we're discussing about aerosols, so different zoonotic infections. So then there are factors which influence the viral transmission, like environmental persistence of virions and viral replication in the body fluids. And then there are the risk factors like the patient's age, the health, the immune status, and the population characteristics. For example, many people may be serum negative. So in those particular people, what we were talking about just now, herd immunity. So suppose many people are actually susceptible to the virus, then a large population will be infected at the same time. Okay. So, and it depends on the geography or the season and ultimately how to prevent and control. Ultimately, what we have known is our own defense. At the same time, there are vaccines. Okay. So vaccines and antiviral drugs we have to prevent and we have to. So we'll see a short video. I hope you understood basically what we were discussing about today. We were discussing about virus host interactions, and we were discussing about different ways in which the virus can enter into the body, and then the way in which our host defense system will act, and it depends on both of them as to how actually it will turn. So we'll see a short video, then we'll sum up. So can one of you just sum up what we have discussed in today's class, so that I know that at least you have understood few things. Yes. Roll number 97, Rupasita. Yes, can you just summarize whatever we have discussed? Just unmute yourself and whatever you have remember, you can say. Okay, roll number 97. Roll number 134. Anyone? Anyone can summarize? Boys, roll number 16. Sarvani, can you just summarize? If you summarize, I'll just understand. If you whatever you remember, if you tell, then I'll know that you have just understood whatever we have discussed. Yes. Anyone? Roll number 33. Sai Durgani. Can you just summarize whatever we have discussed? In short, just tell whatever you remember. Anyone just unmute yourself and say. Either everybody understood everything or nobody has understood anything. Either of the two. So in any case, so I'm leaving the meeting. So thank you all for joining today. Okay.